Plant galls. General Introduction. In this lecture, we will study about what are plant galls, how they are induced by some organisms, and how some organisms get benefited from these plant galls. Let us start. Galls are abnormal growth that occur on leaves, twigs, roots, or flower of many plants. Most galls are caused by irritation and or stimulation of plant cells due to feeding or egg laying by insects such as aphids, midges, wasps, or mites. In general, gall provide a home for the insect, and in these galls the insects can feed, lay eggs, and complete their life cycle. Each type of gall producer is specific to a particular kind of plant. Some galls are the result of infection by bacteria, fungi, or nematodes. Thus, we can define galls as pathologically developed cells, tissues, or organ of plants that have arisen by hypertrophy, means increase in cell size, and or hyperplasia, means increase in cell number as a result of stimulation from foreign organisms. Some galls are induced by virus, bacteria, fungi, nematodes, and mites, but insects are the most gall forming organisms. The study of plant galls is called saccidology. The gall causing animals, insects, mites, and nematodes are called saccidozoa, and galls induced by saccidozoa are referred to as zoosacidia. These plant galls may appear as a ball, knobs, lumps, or warts, each being characteristic of the causal organism. These galls shows different range of color. They may be red, green, yellow, or black in color. Some environmental factors such as weather or plant susceptibility and pest pollution also affect the occurrence of galls on plant from year to year. Oaks are one of the most susceptible, being host to over 500 different wasps, aphids, mites, and midges that cause galls on leaves and twigs. The picture shows leaf of cordia with numerous leaf galls, and these leaf galls are caused by gall mites. The second picture shows Pangamia leaf gall. Most galls are caused by mites and insects. Gall mites, Areophydi, are very small, usually white, sausage shaped mites with only two pair of legs. The three major group of insects that cause galls are aphids and their relatives, gall midges and gall wasps. The aphid group includes aphids, phylloxerans, and psyllids. The aphid genus Pamphigus includes several species that form galls on the leaves, or petioles of cottonwoods, aspens and willows. The genus Phylloxera cause galls on the stems and leaves of pecan and grape. Common galls caused by psyllids, also known as jumping plant lice, are found on hackberry leaves and buds. Gall midges are a large group of small, delicate flies that cause bud galls, blister galls, and galls on leaves and other plant tissues. The most common group of gall makers are gall wasps. Most gall wasps belonging to the family Cynipidae, but a few sawflies and chalkid wasp also causes gall. Gall wasps are most commonly found on oaks, but roses, maples, and other plants also serve as a viable host plants. There is an enormous diversity in the patterns of development, shape, and cellular complexity of insect gall. They range from relatively undifferentiated mass of cell, that is indeterminate galls, to a highly organized structure with distinct tissue layer, means determinate galls. The determinate galls usually have a shape that is specific to each insect species. Cynips, Sacidomites, and Ericococoids form some of the most histological complex and specialized galls. These galls have distinct tissue layers or types that may bear little resemblance to the plant part from which they were derived. Galls can be stimulated to develop only from actively growing plant tissues. Therefore, galls are initiated on young leaves, flower buds, stems, and roots, and rarely they are stimulated on mature plant parts. Some complex galls develop only from undifferentiated meristematic tissues which become molded into a distinctive gall by the activities of the insect. Development and growth of insect-induced galls depend upon continued stimulation of the plant cells by the insect. Gall growth ceases if the insect dies. Various workers have reported that the abnormal growth within the galls are associated with various biochemical changes in the levels of carbohydrate, proteins, nucleic acids, phenols, auxin hormone IAA, indole acetic acid, and oxidizing enzymes. In case of the stem gall of Prosopis cineraria induced by an unknown chalkid, the immature stem gall tissues showed almost twofold increase in the protein content. These biochemical changes altered the differentiation process in the host tissues by proliferation of parenchyma. Tissues near the inducing insect shows cytological and morphological changes that benefits its feeding process and development. This tissue is known as nutritive tissue and it presents higher concentration of sugar, lipids, proteins, nitrogen and other nutrients that provide a continuous source of food for the insect and shows intense phosphatase activity. Typical nutritive cells shows a dense cytoplasm with abundant cell organelles, fragmented vacuoles, a hypertrophied nucleus and nucleolus, and dedifferentiated plastid clustered around the nucleus as well as chloroplast modifying to various degrees and a modified cell wall. There is an advantage of feeding on gall tissues rather than normal plant tissues as insects get availability of high quality food. Galls furnish a more protected microenvironment to sedentary surface feeders such as aphids, psyllids, and cocoids than the normal plant surface. Some Saccidozoa may escape from certain parasitoids and predators that are unable to penetrate galls, particularly galls with thick woody walls. For further readings, I would like to suggest these papers. Student, please take benefit of it. Thank you for watching.